Okay, firstly, thank you very much to the organisers of Enforum for allowing us to present today. It's a really good initiative, despite the current conditions around the world that we are all facing, including some of us who have gone for the COVID cut. It's absolutely required during this time. So thank you once again to the organisers, and hopefully the presentation is interesting for all. Today I'm going to be talking about some novel non-conventional wine yeast from the Australian Wine Research Institute and their impact on sensory characteristics. I've broken the presentation down into two parts of the agenda. The first part of the presentation is about the identification of novel non-Saccharomyces cerevisiae yeast identified at the AWRI, and the second part is about the characterisation of those yeast strains and their sensory attributes. So firstly, where do yeast come from? They come from a wide variety of sources. They've been found on apple trees, they've been found on figs, rotting figs. But importantly for the winemaking process, they can be found in vineyard soils, they can be found on the bark of vines, they can be found on the grapes and on broken grapes in particular. They can also be found in barrels and in other parts of the winery. How genetically diverse are they? Well, they are very genetically distant from each other. They've evolved over time through many, many thousands of years. And the genetic difference between some of the early yeasts that have been identified compared to the later, more industrial type of yeast like Saccharomyces is about the difference between a human and a fish. And there's a lot of, obviously, genetic diversity between those two in terms of the way they appear, the way they behave. And that's interesting for yeast scientists to try and identify novel aromas and flavours and characteristics for the future benefit of the winemaking process. How do these populations change in grape juice? Well, they do vary. Some of these yeast do rise very early in fermentation, but then they die off. They can't tolerate high alcohol and high stressful conditions. Other yeasts just don't reach the populations. They really struggle. They can start early in grape juice, but they disappear over time. Other yeast are able to struggle through the early parts of fermentation, and then they really start to grow and then die off when the alcohol gets higher. And then there's yeast like Saccharomyces cerevisiae, as well as Saccharomyces uvarum, which have become evolved over time and involved in a lot of industrial processes such as bread making, beer making, and the wine making processes that we enjoy today. And why this is important is they've evolved in such a way that they do a couple of very important processes in the winemaking process. So what is their exact role? A lot of people see wine yeast as just a converter of grape sugars like glucose and fructose through the glycolytic pathway into mainly ethanol, as well as other secondary metabolites such as esters and other higher alcohols. But they also play another important function where enzymes in yeast can actually convert non-aromatic compounds in the grapes, such as these precursors, which get cleaved and they get evolved, or they get released from the wines into aromatic compounds like terpenes and thiols. And why that's important is that there's a lot of different yeasts that give different aromas. Saccharomyces cerevisiae is a nice sort of estery, pleasant yeast. Candida stellata, found in many grape juices around the world, can produce acetic acid and ethyl acetate, not so pleasant, but they can accentuate wine aromas, so they can have a particular influence. You've got Hansennia spora, which gives off a real banana ester, and Lanchantia, which gives off more floral and rose aroma-like compounds. And so the way that these yeast appear during fermentation and how many of them are present has a big influence in the way that wine aroma and flavour takes hold during the winemaking process. I mentioned that alcohol can also be influenced, and right around the world, as many people and trends lead towards riper fruit styles and riper wine spectrums, there's been this gradual increase in alcohol production. If I take my home country of Australia as a good example, For about 15 to 20 years there was a very sharp increase in the rise of alcohol concentration in wine as people went towards these riper fruit-style wines. I'm pleased to say that that trend has started to arrest, and there has been a decrease in the levels of wine for a number of reasons. And if we can just quickly look at some of the reasons of why you would want a reduced alcohol content in wine, it's because basically the levels become too much in the palate, and it just becomes too hot and unpleasant in the wine. There's also profitability reasons for wineries, where import taxes into various countries become too high as the alcohol content increases. There's also a lot of society, socio-economic reasons as to why we would want to reduce alcohol content in wine. And then there's the impact on yeast during fermentation, where the alcohol rises too high and it becomes inhibitory, and we end up with stuck and sluggish fermentation. So there's been a lot of research into trying to identify yeast strains that can reduce the alcohol content in wine. It's a very natural process, and it has the least impact on the final wine aroma and flavour. Unfortunately, with a lot of commercial wine yeast out there, it's a very, very small increment of reduction that can take place, somewhere in the range of 0.2 to 0.3% volume per volume. So other methods may need to be looked at to try to reduce the alcohol content of wine. And this is where the AWRI took a range of non-Saccharomyces yeast and looked for some novel aromas, flavours and characteristics that you might be able to reduce alcohol content in wine. So they took 50 non-Saccharomyces strains, looked at a variety of conditions, and in particular looked at this sequential inoculation technique. And just to take you through that, a common process for a Saccharomyces cerevisiae is to convert all those sugars that are in the grape juice into alcohol and other aromas and flavours. The idea of using a non-Saccharomyces is that they may be able to convert those sugars to lower levels of alcohol, while potentially bringing some novel aroma and flavour characteristics to the wine. You then add the Saccharomyces cerevisiae to ensure that that fermentation goes through to dryness and complete. Most non-Saccharomyces do not complete grape juice fermentation to dryness. So that's why you need the Saccharomyces cerevisiae over the top, added about a third to halfway through the fermentation to complete the fermentation process. So the results of using this particular process, using a range of non-Saccharomyces yeast in sequential inoculation, resulted with both aerobic and anaerobic conditions in a variety of different aroma and flavour wine styles, but in particular, using the aeration process, they were able to find yeast that were able to reduce alcohol contents by up to 1%. And increasing that aeration actually decreased the alcohol content further. So this preliminary screen was quite exciting that there might be some yeast of interest. These were then looked at for their volatile composition and whether there was anything significant in the aroma and flavour composition that could be potentially positive or negative. Not going to go through the detail here, but it's very important that a lot of these yeast had very similar winemaking characteristics to the standard Saccharomyces cerevisiae, so some great potential there. When moving into anaerobic conditions, using a sterile Chardonnay to make sure that the yeast that were being inoculated were actually doing the job, we were able to see a 0.9% volume per volume reduction in alcohol, so keeping with the trend of lowering the alcohol concentration. And using a non-sterile grape juice, in this case a Shiraz, using a Metschnikowia pulcherrima strain sequentially inoculated with Saccharomyces cerevisiae resulted in up to 1.6% lower alcohol, which was very exciting. However, because this was a non-sterile grape juice, we needed, or the AWRI needed to analyse whether it was actually the yeast inoculated doing the job, or was there something else in the background? And serendipitously, or by chance, we managed, or the AWRI managed to find this Saccharomyces uvarum hiding in the background in this non-sterile Shiraz juice. Interestingly, when a wild ferment was conducted, this Saccharomyces uvarum was hanging around. So within this Shiraz, there was a Saccharomyces uvarum that looked like it could reduce alcohol by up to 1.6%. Quite exciting. So further experiments were done to really try and analyse this Metschnikowia and Saccharomyces uvarum to see whether they were indeed favourable for aroma and flavour properties as well as the alcohol reduction. Once again, this time in a sterile Shiraz, to make sure there were no other influences in the background, the Metschnikowia and the Saccharomyces uvarum were able to lower alcohol by up to 0.9%. So keeping in trends, maybe not the same degree of alcohol reduction, but nevertheless a substantial reduction in alcohol. Interestingly, when a little bit of a play around experiment was done, when you actually combine those two together at the start of fermentation and then sequentially inoculate with a Saccharomyces cerevisiae, there was a 1.8% reduction in alcohol, which was very, very exciting and certainly worth taking to winemaking trials. So at this stage, some Merlot grape juice was sourced. More winemaking conditions were trialled. This is now in a 30-litre pilot scale winemaking facility. The Merlot was treated with Velcorin to sterilise the grape juice, once again to make sure that the yeast being inoculated were doing the job. There was a Metschnikowia pulcherrima used at the start, sequentially inoculated with Saccharomyces cerevisiae. The Saccharomyces uvarum was actually able to complete fermentation on its own. It didn't need sequential inoculation, and we also did the AWRI did an inoculated control. And the results show that the Metschnikowia inoculation was able to reduce alcohol by 1%. The uvarum, once again, a substantial reduction of up to 1.7%, and the uninoculated control was even 0.7% lower as well. So very interesting. Now, to confirm that these yeast were actually doing the job of fermentation, some metagenomics work was done, and you can see here that the cerevisiae control dominated the fermentation when it was inoculated, despite some varying populations of other yeast in the background at the start, but they did not last. The Metschnikowia ferments, when you inoculated that at the start, definitely dominated the first few days of fermentation, but then died off, and the cerevisiae was able to complete the fermentation. The Saccharomyces uvarum, despite having some mixed populations on the first few days, it did dominate the fermentation. And the uninoculated control was a complete mix of different yeast species throughout the fermentation, up until halfway when a cerevisiae was able to take control. So we've got some good population dynamics here to prove that the results are indeed a complete factor of the yeast that were inoculated. Volatile analysis was then done to determine what was different between these wines that were produced. And you can see here that the Saccharomyces uvarum wines were quite different to the Saccharomyces cerevisiae control wines, whereas the Metschnikowia pulcherrima inoculated ferments seem to be very closely linked from a volatile point of view to the uninoculated ferment. These wines were then taken to sensory, and the sensory panel identified that the Saccharomyces uvarum wines were not very pleasant. You can see here wines very high in vegetal aromas, meat aromas, barnyard aromas. Not exactly what you would want to commercialise and sell as a commercial wine, despite the alcohol reduction. Whereas, and the blue line here is the Saccharomyces cerevisiae control. Interestingly, as noted in the volatile analysis, people were able to sense that the Metschnikowia inoculated wine, sequentially inoculated with cerevisiae, was actually very similar to the uninoculated ferments. There was much more purple tint, much more overall fruit aroma, much more overall fruit flavour, red fruit flavours, and red fruit aromas. So there was a lot of positive characteristics here in aroma and flavour to combine with the lower alcohol production. So this was extremely positive and warranted further investigation. So just a quick summary here at this point of the first part. that non-conventional yeast strains can show some potential to lower alcohol content in wine, both aerobically and anaerobically. Saccharomyces uvarum has a great potential to lower alcohol content in wine. However, the aroma and flavour characteristics are certainly not pleasant and probably don't warrant any further characterisation. However, the Metschnikowia pulcherrima strains, and in particular two, AWRI 1149 and AWRI 3050, showed some very favourable sensory characteristics in this red, the purple tint, the red fruit aromas, the red fruit flavours, as well as reducing alcohol content in wine. And that definitely needed to be investigated further. And that's where AB Biotek stepped in. We were very, very interested in this research. We'd seen a number of posters at various conferences. And so we wanted to characterise these yeast further in winemaking conditions at a commercial scale. One of the challenges we faced was to try and make an active dried yeast of this so that winemakers around the world could use it as a commercial preparation. But these yeasts have very different characteristics. They like aerobic conditions at the start of fermentation, so they're very difficult to grow and make commercial quantities of. Their nutritional requirements are very different. Their physical characteristics are very different. Remember I said at the start that the genetic variation in yeast is very different. Their physiological appearance is very different, and Metschnikowia is a good example of that. We need to develop different drying protocols. It was very difficult to actually get a dry yeast from these particular strains. And they also grow very, very slowly in grape juice. Despite being found in grape juice, they actually find it very difficult to get going and do a grape juice fermentation. Now, I also note at this point, we did try and make the Saccharomyces uvarum yeast that I said didn't produce a very nice wine. We actually can make this yeast. If there's anybody out there that would like to make a vegetal, barnyard, meaty style wine, please let us know. We can manufacture and sell you this active dried yeast preparation for you. And more importantly, with the Metschnikowia, we were able to make the yeast, the AWRI 1149 strain in particular, as a active dried yeast. And we took these wines through a commercial Shiraz and Cabernet Sauvignon. The winemaking parameters are there on the screen. And we also used, in this case, a low dose and a high dose preparation. We wanted to make sure in the high dose that the yeast dominated the fermentation and didn't have any competition in the background at the start of fermentation. We co-inoculated this yeast halfway through the ferment. We know it can only get to about 6 or 7% in some lab assays that we've done. So it needed a commercial Saccharomyces cerevisiae, such as Maravin AWRI 796, inoculated to complete the fermentation down to dryness. And here are some of these results at a commercial winemaking level. Importantly, as a dry yeast, the AWRI 1149 Metschnikowia pulcherrima was able to show a typical fermentation profile. It has this 2 to 3 day fermentation delay, a lag phase, and then it does a certain amount of fermentation and then dies off. I just got some PCR pictures here to show you that at day 5, just before the inoculation of the cerevisiae, the Metschnikowia pulcherrima was dominating the fermentation. So that was a very good sign. Unfortunately, though, you can see here that the alcohol reduction was not seen. And this was a bit of a question. Why did we not see the alcohol reduction? At lab scale, it was very, very clear. Was it to do with the yeast cell numbers? Was it because of our dosage rates? Was the background cerevisiae and other yeast having an influence here because we're dealing in non-sterile grape juices now? Was it the temperature or sulfur additions influencing the Metschnikowia population dynamics and the way it was able to do the ferment? Was it the way that we commercialized the dry yeast? So we did a number of different isolations, and we confirmed that taking a number of isolates from the dry yeast population and putting them through a lab situation in sterile juice, we were able to confirm that our dry yeast preparation, no matter how many isolates you take, is actually the right genetic population. We do have a very fit, healthy, active dried yeast population that can do the alcohol reduction. It just doesn't seem to be as effective in commercial grape juices. So what we wanted to do at this point was to go ahead and redo the AWRI 1149, and this time take the AWRI 3050 along and try and make a better preparation and try a number of different techniques. We changed the dosage rates, we changed the sulfur additions, and we made a few other little winemaking technique changes. And otherwise, everything else was kept very, very similar. Importantly, this time, we saw very similar results in the fermentations, but we were so interested in the sensory characteristics when we smelt the wines at small scale that we wanted to now, instead of necessarily only focusing on the alcohol reduction, we wanted to look at the aroma and flavour profiles. Remember, I said these red fruit, purple tint type characteristics were very prominent with Metschnikowia pulcherrima, the AWRI 1149 and AWRI 3050 strains. And what you can see from this data at the bottom is that things like anthocyanins and tannins are actually dramatically increased with the Metschnikowia pulcherrima ferments compared to some of the Saccharomyces cerevisiae controls like Marovan PDM, Marovan AWRI 796, fairly standard Saccharomyces cerevisiae strains. So the actual attributes of the wine were now starting to take almost precedent over the alcohol reduction property. If I take you to the Cabernet Sauvignon, it's a very, very similar result. Once again, a very similar fermentation profile. Genetics checks were showing a very strong presence of the Metschnikowia. And this time in the Cabernet, we were seeing colour density increasing dramatically with the AWRI 3050. We're able to see anthocyanin increases, and non-bleachable pigments were also stronger. So a lot of clear signals that the purple tint we'd seen at the small scale was coming through in commercial scale winemaking as well. So this was quite exciting, even if we weren't seeing the alcohol reduction. When we look at the volatile analysis, you can see here that the Metschnikowia strains obviously were very, very similar compared to the Saccharomyces cerevisiae controls and the uninoculated control as well. And if you take that further to the sensory results that were seen from a range of, I think it was 20 to 25 people on the sensory panel using a range of different descriptors, there were 6 out of 29 that were statistically significant amongst that panel. And these were colour intensity was higher with the Metschnikowia inoculated ferments. Vegetal aroma was actually decreased. Vanilla aroma was slightly better. The reductive aroma was substantially less. The tannins were increased, and the dark fruit flavour was also increased compared to Saccharomyces cerevisiae wines. So this was looking like a very, very favourable wine in terms of its aroma and flavour properties. When we looked then at the Cabernet Sauvignon, once again, the volatile analysis showed a great deal of variation between the Metschnikowia wines and the Saccharomyces cerevisiae wines. And in particular, when once again we took these to sensory, in particular the Cabernet Sauvignon this time, you're able to see here that the Saccharomyces cerevisiae wines were giving very, very different characteristics. And in particular, the AWRI 3050 strain seemed to give this greater level of dark fruit characteristics, more pungent, more aromas were seen compared to the cerevisiae wines, which may have even been a little bit earthy or rubber-like on the nose. So this was looking very favourable as well. Now, one thing people are asking is, it's looking very good now from an aroma and flavour point of view, but what happened to the alcohol reduction? Was it noted? And what we noted was that when using high doses of the AWRI 3050 strain, there was about a 0.3% reduction in alcohol, consistently, statistically significant across both the Cabernet and the Shiraz. And that is consistent. We are consistently seeing that now in other juices, but unfortunately, we're not seeing that 1%. And there's a number of different reasons for that, we think. And so the AB Biotek research scientists are looking at various ways of trying to manipulate the winemaking conditions to try and make this alcohol reduction to a greater magnitude. So in summary, looking at the 2 years of commercial winemaking that we've done with these particular Metschnikowia pulcherrima strains, we are seeing consistent characteristics in both Shiraz and Cabernet Sauvignon. There are higher scores being seen by sensory panel for positive characteristics, and in particular, we're seeing lower scores for the negative characteristics. We're also getting strong indications that the higher dosage rate of active dried yeast is leading towards an alcohol reduction of about 0.3% volume per volume in commercial winemaking. And we actually did some informal sensory following these trials a little bit later, about 12 months later. And interestingly, all of these attributes are remaining in the wine. So it's not like it's a 3 to 6 month characteristic. It does look like these characteristics remain in the wine and remain integral to the characteristics of the final wine. So overall, we feel that there's a really good opportunity for these 2 yeast strains, AWRI 1149 and particularly AWRI 3050, to have a role in the commercial winemaking process for improving the positive characteristics of the wine aroma and flavour. And this should enable our customers to improve their wine styles, potentially take some of the greener characters out of wine and produce that darker fruit aroma with a little bit less alcohol as well. So overall, this has now led to a commercialised outcome. And there is now a product available called Malvern AWRI Obsession. This product is commercially available from AB Biotek and through our distribution network. This product, AWRI Obsession, does show increased colour. When people tilt the glass of wines made with this yeast, you get a very strong purple right to the edge of the wine. There's no clearing towards the edge. It's very, very intense wines. We're also seeing a lot more complex aromas, a lot more dark fruit, a lot more red fruit is being seen on the nose, and in particular on the palate as well. So it's allowing that riper fruit spectrum to be seen without necessarily having riper fruit to start with. And importantly, it's able to reduce and almost mask some of the green characteristics that can be seen in some grape varieties and from certain regions around the world. So we feel this yeast has a great opportunity to mask green characters, bring out those riper fruit spectrums at a lower sugar level at the start of fermentation. Finally, just some acknowledgements, and particularly from the Australian Wine Research Institute. They would like to thank the Australian Government and in particular the Australian Grape and Wine Authority for helping fund their particular projects. Collaborators such as the University of Santiago in Chile and the University of Rivardi, Virgili in Italy as well. I'd like to thank for the second part, in particular the characterisation work, all my colleagues at AB Biotek, for the Geisenheim Institute for doing some of the analysis, and University Polytechnic de March as well for their assistance. So many thanks to all of our collaborators. Hopefully you find the presentation interesting, and questions can always be emailed to me, or please give me a call on the appropriate time zone. Thank you very much for listening.